In this video, we will discuss about synergetic effect of immune checkpoint blockade and HTPBD with TRAP1 targeting genes and 5FU treatment. So it has been well known fact that when the colorectal cancers are treated with 5FU, then it will cause necrosis, and that seems to be the cause for generating chemoresistance against this 5FU. So since they have already optimized the efficiency of HTPBD plus 5FU, now they wanted to reduce this chemoresistance. So for that, this immune checkpoint blockade could seems to be a perfect candidate, as it will block the receptors that will trigger immune suppressions on the tumor cells. And one of the common receptors is PD1 that is present on the tumor cells. PD1 ligands that is present on the tumor cells. So, and PD1 receptors are usually present on the immune cells. So these PD1 ligands will be neutralized by this anti-PD1, which is a kind of immune checkpoint blockade therapies. So first, what it is, they developed, in order to check their hypothesis, they first developed the chemoresistant cell lines of this CD26, which is the colorectal cell lines. So what it is, they increase the concentration of CD26 with, they increase the concentration of 5FU to this CD26 in vitro and make them drug resistance after like 45 days, and then inject those cell lines into the mice and then use different treatment conditions to check which of them works well. So some of the different conditions are this HTPBD alone, 5FU alone, and HTPBD 5FU, and third condition is HTPBD 5FU plus anti-PD1. And the results what they have seen is that in the case of control, they can see the tumors are increased to a greater extent. When they use anti-PD1, they can see some suppressions in the tumors progression. When they use 5FU plus anti-PD1, they see further decrease in the tumor progressions. And when they use the control HTPBD plus anti-PD1, the tumor decrease occurs, but tumor decrease occurs compared to the control, but not to the minimum level. And when they use HTPBD plus in HTPBD control plus 5FU plus anti-PD1, they can still see some tumors. And when they use HTPBD TRAP1 plus anti-PD1, they can see so far the lowest tumor progressions. And HTPBD plus FU, they see some tumor progressions. And when they use HTPBD TRAP1 plus FU plus anti-PD1, they see a minimum tumor progressions compared to all the other conditions. And when they check the body weight, they can see that body weight also increases with when they use for the control, and body weight also increases for the control and when they use anti-PD1, but it is minimum when they use HTPBD TRAP1 plus 5FU plus anti-PD1. We can see that. And when they check the survival rate, they find out that the last conditions will increase the median of the survival rate compared to the all other conditions. And in terms of this luminescence, there is quantifications of this luminescence that they have observed. They can see that luminescence is minimum for the case where luminescence of the tumors, bioluminescence of the tumors, so it's minimum for the case where they use HTPBD TRAP1 plus 5FU plus anti-PD1. Thereafter, they wanted to check this in vivo effect in the mice. So in this case, they directly take this APC mice models that can develop these colorectal cancers, and then treat them with HTPBD 5FU anti-PD1 and the combinations of this. And what they are able to first we see the tumor results. We can see that tumors seems to be higher, which is marked by black arrows in the control conditions, and also in the anti-PD1 and 5FU plus anti-PD1, HTPBD plus anti-PD1, and HTPBD TRAP1 plus 5FU. But this is minimum. This tumors flow is minimum in the case where they use HTPBD TRAP1 plus 5FU plus anti-PD1. And these things, they have spontaneous tumor number has been quantified in the panel H, where we can see that tumor numbers are minimum for the last condition, where they use all the three reagents together. And in the panel I, they shows the immune cells that are activated, and here we can see that immune cell activations seems to be higher in the bottom row of the panel I. In the that is CD8 plus CD4 plus signals are higher when they use all the three conditions. And in the top figure, they shows that tumors that are present in the small intestines of the mice, which they harvest after the at these 48 days. You can see that tumors is almost negligible in the case where HTPBD TRAP1 plus 5FU plus anti-PD-1 is used. Thereafter, they check the tumor presence using some radioactive reagents, radioactive fluorines. I'm not able to recall its exact name. And they can see that for the control, the tumor signals are higher, but for the HTPBD TRAP1 plus 5FU plus anti-PD-1, the tumor signals are minimum or lower, significantly lower. The quantifications is not shown, but the results are quite visible. So all this indicates that this immune checkpoint blockade therapies can be combined with this HTPBD TRAP1 plus 5FU to suppress the tumor progressions and delayed and increase the survival life of the patient. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.